Thank you so much. It's a tremendous honor for me to be able to speak with you this morning, and thank you for that wonderful introduction. So I feel really privileged and honored and humbled to receive this award. I thought probably the best thing for me to do is tell you how I got here, which really, as Professor Stroma mentioned, was not a very straightforward path. So I grew up in Albany, New York, in the United States, and my dad, he ran a small liquor store, and he worked very hard, but when he came home, he'd play math games with me. And my mom, she was a homemaker. She took care of myself and my sister, and my mother worried a lot. She's actually 99 years old, and she still worries a lot, but she's one of the nicest people I've ever met, and I hope some of that rubbed off on me. You know, I just thought I'd show you a picture of me as a little boy. My parents got me these what were called Gilbert sets, erector sets and chemistry sets and microscope sets. And that, I think, was very good for getting me into science. You know, with the microscope set, I'd watch shrimp eggs hatch, and we had this basement in the house, and I kind of set that up as a chemistry lab where we'd mix chemicals together. I always liked magic, and I liked that you could mix these reactions and they change colors. So that was me as a child. And then in high school, I wasn't very good in classes like French and English and things like that. But I did okay in math and science, and my guidance counselor and dad said, well, if you're good at that, you should become an engineer. And I really didn't know what engineers did. I thought they ran railroad cars. But at any rate, I decided to do that, not knowing better, and I went to Cornell University and majored in chemical engineering. But when I finished, my bachelor's degree, I got some job offers to run chemical plants, but I didn't think I'd be very good at it. In fact, I'm sure I wouldn't have been. So I decided to apply to graduate school, and that I'm sure was very good for those chemical plants or the people who work there. I went to MIT for graduate school, and I got a doctorate in chemical engineering. I still didn't know what I wanted to do when I graduated. And when I graduated in the 1970s, I actually graduated in 1974, there was this gas shortage in the United States, and the prices of gas kept going up and up. And what happened is if you lived in Boston, where I was, you actually had to wait in line to get your tank filled up for your car for 2 hours. It was really amazing. But the good thing is that if you were a chemical engineer, you got a lot of job offers. And so all my friends that graduated around that time, they went to work for oil companies. I thought that's what I'll probably do too, just like my friends. So I went to interviews. In fact, I'd never been, I was 24 years old, I'd never been east of Boston or west of Buffalo, New York. That's pretty narrow. But then I got to have a lot of interviews and see the country, and I got all these job offers. I got about 20 job offers, four from Exxon alone. Believe me, I wasn't that good, but they just had so many openings. One of them, I remember I had one of these interviews at Exxon, and when I was there, this was in Louisiana, and I remember one of the older scientists said to me, he said, if you could just increase the yield of this one petrochemical by 0.1%, he said, that would be fantastic. It would be worth billions of dollars. And I remember flying back to Boston that night thinking to myself that I really didn't want to spend my life doing that. So, well, what did I want to do? So I've always had this dream of wanting to use my education to help improve people's lives. And when I was supposed to be doing my doctoral thesis at MIT, and I did do it, but when I actually spent most of my time helping start a high school for disadvantaged high school kids. And I got very involved in developing new math and chemistry curricula. One day I saw an ad at what's called City College of New York to be an assistant professor to do just that. And so I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea, so I found about 40 ads for different colleges, a lot of them not particularly good ones, but anyhow, I wrote applying for jobs to do just that, and I don't think any of them wrote me back. So that wasn't so good. So I started to think, well, what other way could I use my chemical engineering education to help people? And so I thought about medicine. So I applied to a lot of hospitals and medical schools. They didn't write me back either. But one day, I remember going into the lab, and one of the older scientists said to me, he said, Bob, he said, you know, there's a surgeon in Boston named Judah Folkman, and he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But at any rate, so, and he was kind enough to offer me a job. So I think a lot of people said to him, well, you know, I was actually the only engineer in the hospital, you know, when I went there in 1974. And a lot of people said to him, well, why would you ever do that, hire somebody like that? And they also said to me, why would you go to a hospital when you could work at all these oil companies? But anyhow, I loved that job. That job changed my life, and I learned so much. And the projects that I began working on, they involved two related problems. Dr. Folkman's big idea, which was very controversial, it turns out, but I didn't know any biology, so I didn't realize that. But anyhow, his idea was that if you could stop blood vessels from growing to a tumor, that might be a whole new way to stop cancer. And so one was the first project was to prove that he was right, but in so doing, isolate the very first substances that could stop blood vessel growth. And to do that, one of the key things was developing a way to study blood vessel growth, and all the molecules that you might use to inhibit blood vessel growth or stimulate it, they were large molecules. And if you injected them into the body or any organism, they'd get destroyed. So we needed to develop a way to create tiny particles, like Professor Roma talked about, nanoparticles or microparticles, that could deliver for the first time a large molecular weight substances. Now, before this, people had done a little bit of work on what's called drug delivery, but very little, and always was for small molecules. People always wrote, in fact, Dr. Folkman was an advisor to the only company in the world at that time, a company called Alza, that had done really work on drug delivery, but they always used tiny molecules, a couple hundred molecular weight. And people, when he asked people, including Nobel Prize winners, about helping us, they always said, well, you know, if you try to encapsulate something in a tiny particle, there's no way that the large molecule can get out. It's like any of us walking through a wall. They're just too big. So people told him it was impossible to do. But if we were going to solve the blood vessel problem, the angiogenesis problem, we had to solve the delivery problem. So I spent 2 years working on this project, and I found over 200 different ways to get this to not work. But eventually, I made this discovery that you could make tiny particles and with certain materials. We used polymers initially, and here's a picture of one, and it's cut in half. And you could really make these any size. But and what we were able to show in the journal Nature in 1976 is that for the first time you could release large molecules, and also, as Professor Stroma mentioned, this is actually the first time nucleic acids, which are DNA or RNA, had been delivered from tiny particles. So, and then what we did is we used these systems to create ways to study these angiogenesis inhibitors. So we put, I'd isolate, and this is just an aside, but we put these different molecules that I was isolating in these little tiny systems, and we had bioassays to study blood vessel growth. But you know, we wrote this paper in 1976. We also wrote a companion paper with it in 1976 to the journal Science. This was in Nature, the other was in Science, and that Science paper was the isolation of the first angiogenesis inhibitor. It still took, and this gives you an idea, medicine doesn't move real fast. So the discovery, this was a 1976 paper, both of them, it took 28 years and literally billions of dollars in work by many companies, including Genentech and others, before the first blood vessel inhibitor was approved by FDA. But starting in 2004, something remarkable happened. And this may be hard to see, this slide in the back, but what you see is starting in 2004, 28 years after these papers, one after the next blood vessel inhibitor is approved by regulatory authorities. Avastin, which is the first of these, actually is probably the second most widely used biotech drug in history. It's been used for treating all kinds of cancer. Also, before this, and there are many others that have been approved for other kinds of cancers, as this table shows. And by the way, you could keep seeing these approvals up till today. There have even been 3 approved this past year, as you see on the bottom. But the thing is that not only can you now use these to treat cancer, but if any of you, and I hope you don't, get diseases of blindness in the back of your eye, like what's called macular degeneration or diabetic retinopathy, before this there were no medical treatments for those diseases using molecules. There was lasers that sometimes people used. But now these drugs like Lucentis and Eylea are by far the most widely used ways to treat these diseases of blindness. They're also what are called blockbuster drugs, and they, in some cases, can reverse the blindness. At any rate, going back to drug delivery, after I did that drug delivery, as I was working on the drug delivery project, I also would get asked, as professors are, to give lectures at scientific meetings. So the first time I ever gave a major lecture was in 1976. It was to a very distinguished audience of chemists and engineers at Michigan. And before that, the only kind of talk I had really given was in 8th grade. And that didn't go real well. I remember rehearsing that talk over and over again. It was only a 90-second talk into my parents' mirror in the bedroom. So so what happened is the next day I got up and I gave that talk. I thought I'd memorized it pretty well, but anyhow, I started to give it, and I did okay for the first minute, and then I just could not remember the next word. So I stood up there for another minute, just like frozen, until finally my 8th grade teacher told me to sit down and gave me a not very good grade. It was actually an F. By the way, I brought the speech today, so I won't do that. But anyhow, when the Michigan talk came, I gave it, and that was a 20-minute talk. I kept giving it over and over for several weeks into a tape recorder. And when I gave that talk, I thought, now I'm 27 years old. I thought I did much better. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk, all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them said, "We don't believe anything you just said." And what they said was not too dissimilar to what people had said earlier, that the large molecules were just too big to get out of any little particle. Like I said, it's like we're too big to walk through a wall. And they also said that the solvents we were using, which are called organic solvents, ethanol or methylene chloride, they destroy any of these sensitive molecules. So that was kind of discouraging. And as Maria said, that I tried to receive funding to get grants, and my first 9 grants were rejected. I remember one of the grants that I wrote to the government, NIH, the reviews came back, well, Dr. Lang, we shouldn't fund this. Dr. Langer is an engineer. He knows nothing about biology and even less about cancer. I remember thinking to myself, I knew enough about math to know that was hard to be true, but nonetheless, that's the kind of reviews that I got. And also, I love being a postdoc. For me, being a postdoc as an engineer in the hospital was almost like being a kid in a candy shop. I learned so much. But a lot of my friends said it's probably not good to spend your whole life being a postdoc. So I did apply to faculty positions in chemical engineering. But when I applied, I got no offers. I got interviews a few times, but then what all of them said to me was that this bio stuff you're trying to do, that makes no sense for chemical engineers. They said, you should be doing energy or something else or oil. So I didn't get any jobs. So then I ended up getting a job in the nutrition department. And the reason I got it, the head of the department, he knew Dr. Folkman and knew me, and he was what I'd call a benevolent dictator kind of department head, meaning that since he liked me, he offered me a job, but he didn't bother to ask anybody else in the department what they thought. And that might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and they said I should leave too. And just to tell you a story on that, a few years ago I got another award, and they asked one of my close friends, Mike Marletto, he's a very famous chemist. He was head of Berkeley's chemistry department and later head of Scripps, member of the National Academy of Science in the US. And he was giving a speech, and I just took a little quote from it. It was actually published, and I'll just read this to you. He said, "One evening, I went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist said, quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job." So that wasn't so good either. But actually, I'm still at MIT, so I guess it was okay. So another thing that also that Dr. Folkman once said to me, he said, Bob, you know, when we did this work, we should file a patent. You know, that might seem, when you heard Maria's introduction, that very common that we do that. But you know, in the 1970s, I had 0 patents, and Boston Children's Hospital, which is a very good hospital, they had 0 patents too. So we filed for one for the first time. And 5 years in a row, the patent examiner rejected it. And I remember the people at the hospital coming to me and saying, Bob, you are wasting so much money, just give up. But I don't like to give up. You can probably tell that by now. And so I started to think of other ways to get the patent allowed, you know, legally, of course. And science really wasn't working that much. So I wondered, you know, when I started doing this work, I told you that everybody said that this was impossible, it could never work. So I wondered, maybe somebody wrote that down. So you can do what's called a science citation search, meaning that now we're in 1982 after 5 years of rejections. So I looked back at our 1976 Nature paper to see who quoted it and what they wrote. And I found actually a number of interesting quotes. It's always interesting to see what people say about you. But let me just show you one of them, which was, I thought, very helpful for getting a patent. This is for anybody that wants to get a patent. This is a good thing to know. So basically, these were 5 of the, I had no idea this was written, but these were the 5 of the top chemists in the world, material scientists, and they wrote, describing this field in 1979, that generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One did not expect that large molecules could be released because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so surprising I thought was a really good word for a patent examiner, results that clearly demonstrate the opposite. So I showed this to our lawyers and the people at the hospital, and they flew down to Washington to show it to the examiner. And the patent examiner said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits or written statements from each of these 5 people that they really wrote this. It's a true story. I remember, so I wrote them all, and you know, I was just this young guy, and they were all nice enough to write me back that they really wrote it. And what happened was we got this really broad patent in 1983, and that was the first time that that was allowed. And so then, you know, like I say, I was doing all this work, and I've always to me it was important that what I did wouldn't just lead to scientific papers. I mean, I'm thrilled that what Professor Stroma mentioned in the beginning, that our work has been largely cited by the academic community. But it also means a great deal to me that what we do makes a difference in the world. And certainly helping other scientists, you know, advance fields is great. But I also have always had this dream and passion that the work we do also makes it into the world to help people. And so I was excited that after 10 years of doing this, sort of feeling in the wilderness, that a couple very large multi-billion-dollar companies called me up and said, well, they'd like me to consult for them and try to develop this area. So I mentioned the patents, and they said, well, we want to license them, and we want to give you a grant and a consulting fee, and we're going to put several people in our companies to work on it. And I thought this was wonderful. But what happened was, with these 2 large companies, they did that maybe for a year, but they didn't get exactly the results they wanted, so they gave up. So what happened was, one of my good friends, a couple years later, Alex Klebanoff, said, Bob, we should start our own company. So I got 4 of my students who had worked in this area, and we started this little company called Enzatech, which later merged to form Alchemies. And because of that company, and actually others that licensed our patents or used our principles, today there are all kinds of products based on these concepts and patents that I mentioned. And some of them are shown here. They're probably used to treat over 100 million people every year. They range from some of the most common treatments for opioid addiction, like Vivitrol, to new cancer treatments like Lupron Depot. And see, if you didn't put them in these particles to protect them, they would just get destroyed. But like Lupron Depot, for example, that's developed by Takeda, which is a Japanese company we had an arrangement with. You know, that would get destroyed in seconds. So that little microsphere, that gets injected once every 6 months. But all these end up being used, new treatments for schizophrenia, for pain, drug-eluting stents, which are widely used, probably one of the most common ways of treating heart disease. Another big advantage is if you deliver these drugs locally, like the way the drug-eluting stents work, is you take Taxol, which is a drug that's very toxic. It's an anti-cancer drug, actually. But you can, if you deliver it locally, you can get 1/1000th the dose and keep blood vessels open after heart disease. At any rate, so like I say, a lot of these end up being used. But, you know, we kind of go back and forth. And, like, companies, of course, are creating these products, but we still try to figure out how to solve basic problems in academics. And going back to the MIT lab, one of the things that we saw when we made really tiny particles, like Maria was talking about, nanoparticles, is that they run into several problems. Even though you can get delivery, they aggregate because they're so small, they can stick together like wet sand. And also there's cells called macrophages, and they just eat these little particles. So we came up with a coating with polyethylene glycol. We did this, others have done this too. Vladimir Torchlin was one of the people who did a lot of work with us, and we published this again in Science about 30 years ago. By the way, polyethylene glycol is actually, if anybody has problems going to the bathroom, there's a drug called, or not even a drug, called Miralax. And that, so it's, you know, should be a fairly safe thing. Anyhow, this coating helps prevent the drugs from being eaten, and I thought I'd illustrate that with a video, not one that I made, but Nova, the TV show in the US. They came to our lab once and did some filming, and they explained this so much better than I do, so I'll just show you the video.
It starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should tell you that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But you get the idea. So again, how do you get these to help people? Well, let me just give you one example. In 2010, myself, Derek Rossi, Ken Chen, and Nubar Afeyan had the idea that we could create a company by putting what's called messenger RNA in nanoparticles. As Maria mentioned, we called that company Moderna. There's a dogma that says DNA makes RNA makes proteins. By 2010, protein therapeutics and vaccines had led to many medical treatments, but they could often take a long time. I mean, a classic example is if you have flu vaccine, you take eggs and you grow them up, it takes over a year, and so it's a lot of guesswork as to what vaccine you make against H1N1, H1N2, because it just takes so long. But if you used, so there's a long time manufacturing procedure, but our thinking was if you could take messenger RNA, protect it by putting it in a nanoparticle, inject it into the body is the factory, right? The body does all the work. So then you could just, rather than use the protein as a starting point, you use the messenger RNA as a starting point. And you not only could you do these things much more rapidly, but you could also treat, we thought, diseases you could never treat before, like ones where that could enable new intracellular membrane-bound proteins. So by 2020, Moderna had 13 products in clinical trials, including 8 vaccines. Then COVID happened. And I just want to show you the power of this technology. So this is a real timeline. So the Chinese scientists published the virus genetic sequence on January 11th. It took less than 2 days for the Moderna scientists to design the vaccine. Then, put it in the nanoparticles, manufacture it, send it to our clinical partner at the NIH. And the first patient was tested just about 2 months later. So within another 2 months, this is March, now we'll go to May, we had the initial clinical data. And I think other people at the company thought, I'm on the board of directors of Moderna, but as well as helping start it, but I thought it looked good and it was exciting. But in science, and this is sometimes a sad thing to me, and of course this is not only science but business, a lot of people ridiculed the data. In fact, let me show you the front page headlines of my local newspaper, the Boston Globe. This is now May 19th May 19th of 2020, 2 months after the start of the trial. So the front page headlines is, "This is not how you do science," with my picture on the front page. So at any rate, this sort of little surprising reception. But I want to just go 6 months later. So as I mentioned, I'm on the board, and what we did, and people that follow medicine know probably about clinical trials, but you do phase 1 trial, that was this, then what's called the phase 2 trial, but the key trial is the phase 3 trial. Here we did 30,000 patients, 15,000 with placebos, 15,000 with the vaccine, and it's a double-blind study, so you don't know who gets what. But finally, so this 6 months later, the code was broken, and they told us the results. And I remember sitting in the boardroom on a Sunday, and the results were quite striking. Not a single person in the treated group died. Not a single person in the treated group even went to a hospital, and it was 94 to 95% effective. There was another nanoparticle mRNA vaccine by BioNTech and Pfizer, and that also was quite effective, about 91% effective. Here, just to show you, this is the Moderna vial, and just to show you one example of the clinical data and why people should get vaccines is from the CDC. There are many other things. You can look at New England Journal of Medicine papers, but this is just one time point, and it's not overrepresentative, but you compare unvaccinated to vaccinated with no booster to vaccinated with a booster, and shows the remarkable efficacy of the vaccine. And this will continue, whether it's for COVID or for other vaccines or other therapeutics with messenger RNA. So while Moderna is an example of how basic research from academics can lead to new treatments, we continue in our MIT lab to develop, I hope, new technologies of the future. One example of this is about 10 years ago, I was visited by a very successful software entrepreneur. Let me just show you a picture of him and me in my office. That's me, and there you see the software entrepreneur. For those of you that don't recognize him, it's Bill Gates. And you may know that he's very interested in helping people in the developing world, which we are too. And he asked us, could we help adapt some of our science for purposes like this? So one issue is that people often don't come back, now we know it for the United States, but in Africa, for example, people rarely come back for a second, injection. So he said, is there some way we could develop what we call a self-boosting vaccine, where you might be able to just take one injection and get multiple boosts, like if you look at the graphs on the right. So what we did is we created a new printing process to place vaccines in the center of nano or microparticles. You look at the bottom picture. And then by changing the thickness of the coating or its chemical composition, you think of these as tiny little eggshells. We could make them crack at different points in time, like 1 month, 2 months, a year. So we did that. Anna Jakulenik led that work. And here what you see is one injection with 11 different boosts, at different times. You can program them to be any time frame you want by changing, like I say, the thickness or the composition. We're also working with Mark Prausnitz, who is one of my former students, to put these little self-boosting vaccines in microneedle patches, which a number of places are now working on. And so the idea is when you make these patches, they don't go very deep, so it doesn't really hurt much. And then you could put them they look like little even Band-Aids on the left. And the idea is someday we hope to be able to ship these all over the world, or people could ship them all over the world to treat these diseases. So, another area that I also started thinking about, and Professor Stroman mentioned this, involves creating brand new materials. And working in a hospital, I noticed that almost every material that would be used in medicine wasn't designed for medicine. It was usually a medical doctor trying to solve a medical problem and going to their house to find some material that kind of resembled the organ or tissue they were trying to fix. And then they'd use it in a patient. So in the case of the artificial heart, what happened is they actually, to make an artificial heart, they wanted to have something that had a good flex life. So they asked what object, this is 1967, has a good flex life? And they said a lady's girdle. So that's what they picked, the material in the lady's girdle to make the artificial heart. But when you start down that path, that from an FDA or regulatory standpoint, it's very hard to change. So now we're 55 years later, and the artificial heart's still made out of the lady's girdle. And one of the big problems is that when blood hits the surface of that, the lady's girdle material, that forms a clot, and that clot goes to the patient's brain, they get a stroke and they die. But if you think about it, something was designed to be a lady's girdle, it may not be the optimal blood contacting material. Breast implants, for example, one of those is a mattress stuffing. So I started thinking that, maybe being a chemical engineer, rather than go to your house, you could do chemical engineering design. Ask what you really want in a new material from a chemistry standpoint, biology standpoint, and engineering standpoint, and then design it from scratch. And I remember when we first came up with this, a lot of people criticized it, but I had some terrific students, and we developed a number of new degradable materials this way, one of which has led to a new treatment for brain cancer that's been used for now the last 26 years in over 30 countries. And the last sort of scientific point I wanted to mention, it really goes back to what Professor Stroma mentioned in the introduction. Rather than use this kind of approach, take materials, what if we could combine materials with cells to create new tissues and organs from scratch? This is a paper in Science that we wrote that was been cited over 8,000 times now, and it really details that you could take any cell type, including stem cells, and make them into one of these, put them on a certain type of scaffold, and I won't go into all the science, but you could make virtually any tissue or organ. So, at any rate, today, this concept is kind of the basis for making, as Professor Stroma mentioned, many tissues and organs on a chip. Let me just show you a couple of quick examples. If somebody gets burned, you can now make artificial skin. I'll illustrate that. Here's a little boy who's very badly burned, but you can take the product, which are neonatal skin fibroblasts, put it in the scaffold, and it looks like this. You can cryopreserve these. You can put it in the child at the time of injury. If you come back 3 weeks later, he's doing better. If you come back 6 months later, he's pretty much healed. And these are now approved by FDA for diabetic skin ulcers and also burns. And also, you can make organs and tissues on a chip, which may someday, I believe, really reduce animal testing and human testing and enable you to do what I call high-throughput analysis for new drug development. Just to illustrate that, a couple of my students, Gordana Vunyak and Milika Ritzik, professors at now Columbia and Toronto, made a heart on a chip, starting in our lab many years ago. And you can take heart muscle cells and you can put them on these scaffolds and it'll have the right structure. You can even take a video, and let me show you the video of a heart, 6 million cells on one of these chips, but they'll beat as one. We're not shaking this either, but you can see it beating. And you can even take a EKG of these, or you can use it to do drug testing. And I just, on this last technical slide, was just going to show you that we're working on many different ones. Professor Stroma mentioned that you could do spinal cord, but we've also worked on cartilage, gastrointestinal tract, hearing loss, brain, heart, with Julie Andrews on vocal cords. I mean, she's not doing the science, but at any rate, she's been a big supporter. And so someday I hope you'll see many of these helping either people or accelerating drug discovery. So finally, I just wanted to say a word or two about my personal life. I've been very lucky. I've been married for over 34 years to my wife, Laura. I met Laura. She was a roommate of one of my students, and I'd seen her running in the track where I also ran for exercise. And I've been really lucky. She's not only my wife, but my best friend. And she's also a very straightforward person, so I know exactly where I stand all the time. And I also have three wonderful children. Michael, who's here, and his best friend Livia is also here. Susan in the middle. She's not able to be here. She just gave birth to our first grandchild on Friday, and last Friday. And Sam, who's there, and Sam and his wife Ria are also in the audience today. And so I'm delighted that they were able to accompany me here. And finally, I just feel, so lucky that I've had such a great staff, people working with me at MIT, fantastic students, postdocs, and collaborators. I've seen over 1,000 of my students get great jobs, and approximately 400 are professors around the world. And I view them, my students and postdocs, as kind of like an extended family. I'm really proud of them. And one person gets an award, but it really, it's really all of them that deserve that, and that they've given me so much support and have done such great things. So I just want to say finally that I'm really so honored to receive this award and to have had this wonderful opportunity to share my research, my struggles, and my dreams with all of you. Thank you so very, very much.